Hi everyone, my name is Veroni and my teammates are Ushra, Maria, Hoyan and Ulla. Today we will be talking about the Hippo pathway. The Hippo pathway is a crucial signaling cascade involved in regulating cell growth, cell proliferation, cell differentiation. Dysregulation of the Hippo pathway will result in diseases such as cancer. The Hippo pathway was first discovered in Drosophila fruit flies and then later on in mammals. From 1995 onwards, key discoveries about the Hippo pathway have revealed its role in controlling organ size and then its impact on cellular processes. In 2007, two vital transcription co-activators were discovered, YAP and TAZ. These were identified as central regulators of this pathway.
The Hippo pathway consists of 4 key proteins. First, MST12, which act as sensors, detecting signals like cell density. Once activated, they phosphorylate LATS12, the co-regulators of this pathway. LATS12 then phosphorylate the transcription co-activators YAP and TAZ. These 4 proteins work together to regulate cell growth, with MST12 and LATS12 controlling whether YAP and TAZ are active or inactive. Once YAP and TAZ are phosphorylated by LATS12, they are retained in the cytoplasm, preventing them from promoting cell growth. In their unphosphorylated state, YAP and TAZ move to the nucleus and trigger gene expression that drives cell proliferation. This balance between active and inactive YAP and TAZ ensures controlled cell growth. When the pathway is disrupted, it can lead to excessive growth, contributing to cancer development.
My name is Ulla, and I will be talking to you about the Hippo pathway mechanism of action. The phosphorylated MST protein kinase, along with the non-catalytic scaffold protein salvador, in other words SAV, catalyze the phosphorylation of the MLB accessory protein, which in turn binds to the LATS protein kinase. The MST-SAV protein dimer catalyzes phosphorylation of the LATS protein kinase, which in turn phosphorylates both YAP and TAZ transcriptional co-activators. In their unphosphorylated states, YAP and TAZ enter the nucleus freely and function as transcriptional co-activators for genes that code for mitosis, such as the cyclin E gene, which drives the cell from the G1 to the S phase. In their phosphorylated states, however, YAP and TAZ are deactivated and can no longer go into the nucleus. They either undergo cytoplasmic retention or degradation. This results in no transcription of the genes mentioned. Therefore, it is a stop signal for mitosis in the cell. And this is how the organ size is controlled through the Hippo signaling pathway.
The Hippo pathway is disrupted when there are changes in key parts like MST12 and LATS12, which usually help control cell growth, by blocking YAPTAZ proteins. These changes, often caused by mutations, allow YAPTAZ proteins to become more active. External factors like physical pressure on cells, lack of proper contact between cells, or high density can also weaken the pathway. Additionally, when pathways like EGFR are overactive, they can send stronger signals for cell growth, further disrupting the Hippo pathway. So when the Hippo pathway is disrupted, YAPTAZ proteins move into the cell's control center, which is the nucleus, and turn on genes that make cells grow and divide too much. This can lead to development of cancers where cells multiply uncontrollably, as well as fibrosis where there is too much tissue buildup. In some cases, it can also cause organs to grow abnormally large because the pathway's normal role of keeping cell growth in check is lost.
Hi, my name is Hoyan, so I'm going to presenting about some of the therapeutic applications. The first one is the EFGR targeting therapy. It's a type of cancer treatment that focuses on blocking the activity of the EFGR. By combining the EFGR targeting therapies with drugs that inhibit YAP or TAZ, can effectively control YAP or TAZ overexpression, which is often linked to cancer progression and resistance to treatment. The second one is YAP or TAZ expression inhibition. There are 2 key strategies to reduce YAP or TAZ expression in cancer, which involve microRNA interference and small molecules inhibitors. miRNA acts as tumor suppressor by promoting the degradation of YAP or TAZ mRNA or blocking their entry into the nucleus. Small molecules, on the other hand, can either degrade YAP or TAZ proteins or prevent their nuclear translocation. The third one is YAP or TAZ-TAD interaction blocking. It is a strategy for cancer treatment. Researchers are developing small molecules and peptides that prevent YAP or TAZ from binding to TAD, which halts the activation of these growth-promoting genes. By disrupting these interactions, the aim is to stop or slow the growth of tumors driven overactive YAP or TAZ, making it a key target in therapeutic research for YAP or TAZ-related cancers. In conclusion, the Hippo pathway plays a crucial role in regulating cell growth, proliferation, and apoptosis, with its dysregulation contributing to the development of various cancers. By targeting key components of this pathway, such as the core kinases MST102 and LATS102, and effectors like YAP or TAZ, novel therapeutic strategies are being developed to suppress tumor growth. Approaches like EFGR targeting therapy, inhibiting YAP or TAZ expressions, and blocking YAP or TAZ-TAD interactions show significant promise in controlling cancers driven by Hippo pathway malfunctions. So that's all from our groups. Thank you.